my colleague, Stéphane Auger, who is a faculty member in our Faculty of Pharmacy. He's also the co-director of PRIME, and he will introduce our next speaker. Stéphane, take it away.
Thank you, Shana. Good afternoon, everyone. So it's my pleasure to introduce our next speakers for this afternoon. First, we'll have Pieter Cullis. Dr. Cullis is a professor in the Department of Biochemistry and Molecular Biology at the University of British Columbia. He's also the scientific director and CEO of the Nanomedicine Innovation Network. He is also the founding director of the Center for Drug Research and Development. His laboratory has been responsible for fundamental advances in the generation, loading, and targeting of liposomal systems for intravenous delivery of conventional and genetic drugs. This work has led to 2 products that have been approved by regulatory agencies in the US and Europe for the treatment of cancer. And he is the founder of at least 4 different biotech companies that I'm sure he's going to briefly talk to us about. So Dr. Cullis, thanks for being with us today. And you are on mute still.
Okay, can you hear me? Can you hear me all right?
Yes.
Okay, and you can see my screen. Okay, yes. So yeah, thanks very much for the introduction, Stéphane. I'm going to talk for, I hope I can pack it all in 20 minutes, about some of the work that we're doing here in British Columbia. And my title is BC, an Emerging Biopharmaceutical Powerhouse. I can contend that there is a good deal going on here, as I'll try to illustrate. There is certainly a lot of energy that is going into the biopharmaceutical, biotech, life sciences technology area, and it's an extraordinarily exciting time, as I'll try to point out. I have a few conflicts of interest that I'm indicating here. Let's see if I can get my pointer. I'm a founder of Precision Nanosystems, Acuitas Therapeutics, and Molecular U, which I'll mention precision and Acuitas in the course of this talk. Okay, so just a word or two about BC life science companies and resources. There's more than 1,000 companies that have employees in the life sciences community in BC, employing more than 17,000 people, generating $5 billion in revenues. And some of the largest public biotech companies, Aurinia, Zymeworks, et cetera. And of course, Stem Cell Technologies is still a private company, but is Canada's largest biotech employer. There's been more than $1.5 billion raised in this sector over the last couple of years. So we have large strengths in the areas of antibodies and nanomedicines. I'll be talking quite a bit about the nanomedicine area, but also regenerative medicine, precision medicine, vaccines, gene therapies, and medical devices. And there have been major contributions, as I'll illustrate, to the global response to COVID-19. Now, a lot of this has originated with UBC as a center, and has resulted in many of the spin-out companies that you see in this circle here. But it does point out the role of a major university in generating these enterprises is absolutely essential, and has been really played as I say, UBC has played a terrific role in this process. Just a word or two about Accelera. That was the brainchild of Carl Hansen, who's on one of our panels subsequent to these talks. Quite a remarkable achievement. Not only raising $500 million just before Christmas, but really the first COVID-19 antibody treatment to reach the clinic in North America. And as was pointed out here, from patient sample to human testing in 90 days, using a microfluidic approach to select the most potent antibodies in record time, and then getting very rapidly from that into the clinic. So really quite a dynamite technology in terms of rapid response, is having a significant effect, as is indicated here. as a shield against COVID-19 hospitalizations, infection, and death. So 78% reduction in hospitalizations, prevents COVID-19 in nursing homes by up to 80%, and prevents death, of course, and is currently approved in 8 countries. So quite a huge achievement that has obviously major repercussions in terms of a vibrant biotech community. The Precision Nanosystems, which is one of the companies that I co-founded back in 2010, which is really is originated from a manufacturing point of view of being able to manufacture lipid nanoparticles that have a number of applications, as I'll illustrate, but has now expanded into the full range of potential therapeutics using this technology, and has been funded to the tune of about $45 billion from the federal government to establish rapid response pandemic vaccine manufacturing capabilities. So we're starting to build up a strong resource in this area. Cullis Therapeutics, again, a company I co-founded. This is a slide just indicating that Cullis is now the premier lipid nanoparticle technology provider globally to advance particularly nucleic acid therapeutics, and as I'll point out, has played a major role in the development of the Pfizer-BioNTech COVID-19 vaccine. So an overview is that when I say BC inventing the medicines of the future, there is a bit of the Wayne Gretzky approach here, trying to go where the puck is going to go as opposed to where it is. And this is what I mean by the 3rd generation of pharmaceuticals: 1st generation small molecule drugs, 2nd generation biologics, particularly monoclonal antibodies, of course. But the 3rd generation is really smart medicines that rely on nano-sized drug delivery systems. And I'll indicate their importance. First of all, BC is a global leader. We've really enabled 7 of the total of 13 FDA and EMA approved nanomedicines. But the objective is to deliver small molecules more specifically to where they need to go, and enable large molecules based on DNA and RNA to be used for gene therapies. Now, we're currently faced with what I term a totally ridiculous situation where we have somebody with cancer and we give them a drug that goes all the way through their body. And so you can do the calculation, but most of the time, less than 0.001% of the drug will actually get to the tumor to affect the disease. And of course, the other 99.99% goes elsewhere and causes all sorts of nasty side effects. So the objective here is to use drug delivery systems that go much more precisely to where they're needed. And I would suggest 50 years from now, we'll look back and to this time and we'll say, my God, you mean that we actually used to give drugs go everywhere in the body except where they're needed? The notion of getting much more targeted formulations is an obvious one. The other one is that the reason that we've been confined to small molecule drugs for the last 100 years is really because they're the only drugs, the only molecules that can get inside cells. And so therefore we haven't been able to use the biology of the cell, and particularly the molecular biology of the cell, to treat disease. Now that is changing. And again, drug delivery systems are completely key to that, so that we can essentially have systems that are able to not only deliver molecules more precisely to where they're needed, but also have this capability to deliver the material inside the target cell. And so thus enabling gene therapy approaches. And so this is again a major change in the possibilities of medicines. So what is gene therapy? If you look it up in Wikipedia, you'll see a definition is that gene therapy is the therapeutic delivery of nucleic acid into a patient's cells to treat disease. There's an area of really unbelievably intense interest, because most diseases can potentially be treated using this approach. Now, we do require, as I just emphasized, a delivery system to deliver the nucleic acid polymer into a patient's cells. The unmodified systems are rapidly degraded. They don't preferentially accumulate where you want them to, and they can't get into cells even if you get them there. So most efforts to date have used viral delivery systems as vectors to deliver genetic information, but these have tremendous limitations. They can only be used once because they're immunogenic, they're often toxic, very expensive, etc. Historically, non-viral systems have been too toxic and and relatively impotent, but this is changing and changing very rapidly. And so BC companies are really at the forefront of enabling or using lipid nanoparticle delivery systems that enable a wide range of gene therapies, as indicated by the cartoon here. Okay, so currently, nanomedicine companies that are based in British Columbia are the global leaders in developing these non-viral systems. There's 4 companies indicated here: Cullis, Evonik Laboratories Canada, GeneVance, Precision. They're employing nearly 400 people in the local community. And the work of these companies has led to 2 recent drug approvals by the FDA and EMA and other regulatory agencies. In 2018, a drug called Onpattro to treat a disorder known as transthyretin-induced amyloidosis. And in 2020, the Pfizer-BioNTech COVID-19 vaccine, BNT162b2. It has another name which is very long and unpronounceable, but this is the vaccine that's the Pfizer-BioNTech product. Okay, so just with regard to systems to treat hATTR amyloidosis, a very simple hypothesis, and this is actually something that is prevalent in the area of gene therapy. It's very straightforward to come up with a hypothesis to actually treat a condition. And here, the amyloidosis is caused by the production of mutant transthyretin in the liver, which forms fibrils in the circulation, which cause in turn cardiotoxicity and neurotoxicity. And so the pretty obvious solution is to silence the gene in the liver, in this case using short interfering RNA to inhibit production of the protein. As you can see here, it's a very nasty disease, fatal within 5 years, 50,000 people worldwide suffering from this. So the simple hypothesis is to deliver siRNA to silence this gene to hepatocytes, silence the gene, stop production of the protein, reduce and eliminate production of circulating fibrils, and thus stop the deposition of fibrils in the nerves and hearts, and possibly reverse accumulated damage if you really lower the levels of circulating transthyretin in the blood. But of course, you need a delivery system to deliver the siRNA to the liver and into hepatocytes. And so this is the process that Biopharmaceutical and Cullis Therapeutics developed in collaboration with a company in Boston called Alnylam, an siRNA company, and this is during the period 2005 to 2012 to treat this condition. So taking this oligonucleotide, packaging it into an optimized lipid nanoparticle, and then infusing that into patients. And so this is the task that we set ourselves back about 1995, actually, delivering material to the liver, to hepatocytes, and then getting not just to the liver, but into hepatocytes and into the cytoplasm of hepatocytes. So this is the results of a clinical trial that were announced in September of 2017, where as a trial to ameliorate the condition, the transthyretin-induced amyloidosis, the neuropathy impairment score was the primary endpoint, and obviously with a p-value of I'd like to say 1 over Avogadro's number, but 10 to the minus 24, you know for sure this drug works and extraordinarily well. It actually reverses the condition, and so quality of life indicators, muscle strength, et cetera, are not only better than the placebo, but better than when these people entered the clinical trial. And so really by developing an optimized LNP system to deliver the siRNA to the liver, produced an essentially curative therapy for a previously fatal disease. As a result of this, of course, this was approved very quickly by the FDA in 2018. Similarly, a hypothesis for a gene therapy type hypothesis for a vaccine is also relatively straightforward. And this is where we move to the to the treatment of COVID-19 or a vaccine for COVID-19. Here, the potential solution is to express the viral spike protein in muscle and immune cells by delivering the mRNA to the cytoplasm of those cells, thereby engendering an immune response and therefore having a protective effect. And so we have to do here is deliver mRNA coding for the spike protein into muscle and immune cells, have the cells make the spike protein and present that to the immune system, and then the immune system generating an immune response to the spike protein and therefore to the SARS-CoV-2 virus. So obviously this is rather an important endeavor. As Shana was mentioning, the COVID-19 pandemic has caused considerable worldwide damage, 2.5 million deaths, $10 trillion of economic damage. And this is just some of the stats that we're all getting increasingly familiar with. So for this, we need a delivery system to package the mRNA into an optimized lipid nanoparticle and then go through the normal vaccination procedure. So this was developed here in Vancouver by Cullis, working with BioNTech and Pfizer as a vaccine. And so taking the mRNA and packaging it up in these optimized systems, and then having the viral protein expressed in muscle cells and in dendritic cells, leading to an optimized and actually very potent immune response. Now, as you're aware, last November, a phase 3 trial was concluded of this vaccine. It met all primary efficacy endpoints, 95% effective to prevent COVID-19. And this is 28 days after the first dose, consistent across age, gender, race, etc. Relatively safe, well tolerated across all populations. And I should say 43,000 participants. This is probably one of the most extensive vaccine trials ever done. In many cases, it's difficult to recruit that many people, even if you do a trial over many years for a particular vaccine. No serious safety concerns. The companies are now scaling up globally. They were indicating 2 billion doses or 1.3 billion doses by the end of 2021. And now we're going to be hitting, I think, considerably above that with 2 billion or more. So this has been approved by a great variety of agencies. So the point I would make here is that these vaccine applications are just the tip of the iceberg. There's a multitude of gene therapies. Certainly HIV vaccines are coming into focus. A paper published yesterday would support that. Zika, universal flu vaccine, the list goes on and on. Certainly treatments for cancer, heart disease, and Alzheimer's, when in the same way that I mentioned that there's relatively straightforward approaches, you can actually use the molecular biology of the cell to treat many, many conditions. You can use the liver as a bioreactor to produce therapeutic proteins. Just administer these systems intravenously. The protein is produced in the liver. And you can, as an example, produce antibodies for passive vaccination approaches, ranging from cancer to infectious diseases and many more. The range of possibilities is huge. The other point here that I'd like to note is the extremely rapid and precise way that these gene therapies can be devised and implemented. Once you've identified the protein you want to silence or express or edit, you can produce the relevant siRNA or messenger RNA in a time on the order of, say, a month or so, encapsulate it, and you can have a new medicine available in a time on the order of a month or two. So this is obviously has revolutionary potential. It also has interesting implications for our regulatory processes as to how we bring these new medicines into the system. So just to close, that gives you a bit of an overview of British Columbia with a couple of examples. as an emerging biopharmaceutical powerhouse. Certainly with a large infrastructure now of companies in the biotech area with major strengths in the areas of antibodies, nanomedicines, vaccines, etc., and having made major contributions to the global response to COVID-19. So with that, I'll close. Thank you.
Thank you very much, Dr. Cullis, for this quite impressive overview of the British Columbia ecosystem.